Today we'll be talking about CRISPR-Cas9. What is CRISPR-Cas9? It stands for Cluster Regularly Interspaced Short Palindromic Repeats. The process is to identify a sequence of DNA that needed to be altered using a component called CRISPR. Then an enzyme called Cas9 comes in to remove and replace it with another DNA sequence of interest. The current state of knowledge is to replace a faulty gene with a healthy one and make it behave differently in a positive way. For examples, it is to prevent human genetic diseases. Mosquito DNA can be altered to prevent malaria from spreading and to develop soybean plant to increase vegetative size. Why is the topic of interest? Scientists are looking to determine if CRISPR-Cas9 can be trusted to prevent HIV, cancer, and other genetic diseases on humans while also improving the quality of DNA on human, plants, and animals. Other countries that are using CRISPR-Cas9. In November 2018, a Chinese doctor, He Jiankui, claimed that he manipulated the embryo with a gene editing technique on the twin girls to make them immune to HIV infection. The result remains unknown.
CRISPR-Cas9 experiments, editing of BSV. Banana streak virus is a plant-derived disease that causes badnavirus, which affects the production of bananas. The issue is presence of the integrated endogenous banana streak virus in the B genome of plantain is a major challenge for breeding and dissemination of hybrids. The solution: in order to inactivate the eBSV, the virus sequence must be edited. What was the study? CRISPR-Cas9-based genome editing technology was applied to inactivate eBSV sequences in the host plantain genome. The genome edited events of plantain were created with changes in targeted sites of integrated eBSV sequences in the host genome. What were the results of the experiment? Out of 8 edited events tested, only 6 remained asymptomatic in comparison to the controlled, unedited plants under water stress condition. Therefore, 75% of the tests confirmed inhibition of eBSV. Significance of the experiment: a banana's lack of genetic diversity has put it at risk of extinction due to BSV creating major challenges for breeding and dissemination of hybrids. Therefore, the necessity to design a strategy to irreversibly silence the eBSV in the B genome has become crucial.
CRISPR-Cas9 use on pigs. Simone Lilico, a geneticist, has been working on a way to keep pigs from catching a viral disease called porcine respiratory and reproductive system. Causes pneumonias, stillbirth, and abortions. This virus infects pigs' blood cells by clinging to a surface protein called CD163. Simone Lilico decided that by using CRISPR-Cas9 gene editing in zygotes, they would be able to generate pigs that were missing CD163. After the pigs were in good physical shape under normal farming conditions, they secluded a selection of monocyte and macrophage cells from these pigs and found them to be completely resistant to PRRSV infection. Any controversies? Genes can be edited in early human embryos, which also may affect the descendants. This may be viewed as unethical, unsafe, or ineffective. Future directions. Scientists are still doing further research on the technique and looking forward to utilize this method to prevent serious diseases such as cancer, HIV, and other genetic diseases on humans, as well as seeking for better genes on breeding animals and producing better quality on plants.